going on? B to the Ryan. This is the gut microbiome presentation, and start off with the abstract. So the importance of the gut microbiome is that it aids in digestion. Our question was, how does diet affect the microbiome of two dogs? And the hypothesis is that if the diet is different, then the microbiome of the organisms will also be different. So we compared two dogs, numbers 1002 and 1007. Dog 1002 is an 11-year, 5-month-old miniature Dachshund and dog 1007 is an 11-year, 8-month-old Dachshund Beacon. Both are relatively the same weight and in very good health. Dog 1002 has a consistent diet of farina dry and meow mix, which is cat food, and dog 1007 has a diet consisting of blue buffalo dry and Beneful wet, which is dog food. Moving on to the introduction, what exactly is a microbiome? So a microbiome is the types and proportions of microorganisms living in a specific location. And a gut microbiome, like the microbiome, is the community of microorganisms found in the gut of an animal. The microbiome contributes to many health conditions as well as the overall health of the animal. So continuing on, there is a relationship between gut microbiome and irritable bowel syndrome. So when the gut microbiome is stable, it is able to aid in digestion without any trouble. In recent studies, it has been found that when the gut microbiome of an organism is unstable, different forms of irritable bowel syndrome are the result. The relative abundance of the specific families affects the stability of the organism's stool. Too much of one species of bacteria results in irritable bowel syndrome. This relationship is causal, and we found this from Bolin, 2018. There is also a relationship between the gut microbiome of an organism and autism. So those conducting this research use DNA sequencing methods to analyze the atypical pattern of feeding and nutrition in autism and how they interact with the gut microbiome. So they also selectively surveyed results from rodents to see the impact of the microbiome on the neurobehavioral development of the mice. So the relationship is correlative. They are looking for patterns and seeing whether autism is correlated with a specific gut microbiome ratio. We found this from Ewell, 2013. Okay. So how we conducted this experiment? First, we kind of isolated the DNA and purified it. It was extracted from 2 fecal samples using the Quiagen kit. No changes were made to the protocol here. Then we concentrated the DNA using the Quiagen kit and then the Zymo spin column. To quantify the DNA, we used the NanoDrop 2000 software. This was used to determine the concentration of DNA in each sample. A 260-280 ratio was used with the protein to DNA ratio. Following that, we did a polymerase chain reaction. It was amplified through PCR reactions. Following that, we did a PCR cleanup using an AgentCore Ampure XP kit, which was used to remove leftover buffer and unused primers from the PCR reaction. To index the PCR reaction, we used a unique set of short sequence tags that were added to the ends of amplicons from each sample in order to tell which sample is which. So following this, we did the gel electrophoresis, and what gel electrophoresis tells you is that you're going to see DNA if the bands show up. So we pipetted the samples into a well in the gel, and we ran it at 250 volts for 5 minutes, and this allowed the DNA sequences that were different lengths to go towards the end of the gel, and this showed the desired DNA was present in each sample. And again, there were no changes to the protocol here. Okay, moving on to results. So the concentrations listed here are in nanograms per microliter. So sample 1002, 1007 is here. 1002 had a 9.9 nanograms per microliter, and 1007 had 84.9 nanograms per microliter, sorry. And the 260-280 ratio here was 1.96 for sample 1002 and 1.88 for sample 1007. Okay, at this point we moved on to 2 different animals because the kit wasn't working at the end, so we couldn't get the actual results. But these two animals are two huskies, and they had the same kind of health and similar diet as the two we did previously. So the total number of reads for sample 307, for the kingdom it was 2,022, the order was 1,983, and the genus was 1,680. The total number of reads for sample 308 was the kingdom 17,852, the order is 16,604, the genus is 14,856. The top kingdom classification results from sample 307 was bacteria was 2,022 reads, and the unclassified here is 650 or 665. And unclassified is just we don't know what it exactly is. The top kingdom classification results for sample 308, bacteria was 17,851. It was unclassified 176 reads, and there was one read of a virus. So moving on to the next results, the top order classification results for sample 307. And this is when you kind of see the list of bacteria here for the order. So the Clostridiales are 934 reads, unclassified was 748, Bacteroidales was 546, Fusobacteriales was 255, Erysipetalchales was 71 reads, Coriobacteriales was 32, and Enterobacteriales was 23, and we had Gemellas of 20 reads. The top order classification results for sample 308. Again, Bacteroidales was 7,850 reads. Fusobacteriales was 3,982 reads. Clostridiales was 3,054, excuse me. Unclassified was 1,424. Flavobacteriales was 708. Burkholdeриales was 393. Erysipetalchales was 143. Campylobacteriales was 105. Alright, the top genus classification results for sample 307, we had unclassified was 1,007. That was the most reads for the genus. You get less reads for each bacteria as you move along, so you expect more here and less down at the genus because it's kind of closer to the bottom. So you can see the numbers again. Bacterioids was 336, excuse me. Clostridium 321. Fusobacterium was 171. Prevotella was 166. Sarcina 124. Blottia was 102, and Facilicalibacterium was 92. Moving over to sample 308, you can see it's unclassified 3,172. 5,609 bacteria were red. Fusobacterium is 1,879. Prevotella is 1,511. Cetobacterium is 1,008. Phascolactobacterium is 941. Faecalicbacterium is 880, and Flavobacterium is 679. So this is the results of our gel electrophoresis. You might not be able to see from that far away, but we have the samples listed here. This goes back to our previous one with sample 1002 and 1007. So each sample we had bands present, which indicates that there's DNA in what we extracted. So the DNA extraction was successful. The concentrations were high enough for both samples that we were that indicated that the DNA is pure enough and showed more accurate results. The gel electrophoresis, as I said before, was successful. The bands were present in all groups except for the negative control, which is expected. The thicker bands indicated higher amounts of DNA, and in the negative control, it was 0, but any value would indicate contamination of your what you extracted. The gut microbiomes of the two animals are similar. Many of the same bacteria were found in each level. An animal in sample 308 had evidence of a virus, but the reads weren't high enough to adequately give them a diagnosis of illness or anything. You have to have 1,000 reads for it to be considered accurate, or not accurate, but to use it as a good amount of bacteria. At the genus level, in both sample 307 and 308, bacteria were found. These bacteria are usually beneficial to the organism, but they're found to be detrimental when they're found in other areas of the body. And the purpose of this experiment was to test whether differences in diet affected an organism's microbiome. The results of the experiment conclude that our hypothesis was refuted. The differences in the gut microbiome were not as drastic as we had hypothesized, and therefore, the two organisms' fecal composition, specifically the bacteria, were similar. But the reads from the sample from organism 307 were smaller compared to 308. And here's our citations, and that concludes the presentation. Thank you.